Should we say these problems? We're live. We're live.
Okay, so number 1. So they're kind of written out of order, but like deal with it. So A, it's written the same way the sequence was written on the instructions. We put 3' over here and 5' over here. B, again, just mimicking the original template or the original strand given to us. Sequence, that's what I'm wanting. And then 5' to 3', and then C, taking the original and just changing all of the U's to T's, and it's the same 3' to 5'. And we did that based on, if you look at this beautiful picture, so our template strand is the bottom, coding strand on top, and the mRNA is in the middle. The mRNA is created off of the template strand, and the template strand is read 5' to 3', but then it starts synthesis over on the 3'. So where the 3' is, that's when the 5' end of the mRNA starts. And the notes talk about, or the question talks about, that the mRNA strand was created from the right to the left. So that means the right side of the sequence is the 5' and the left side is the 3'. And then just using that to figure out everything else, and that's it.
Okay, moving on to number 2. All right, so these are the fill in the blank questions. A says blank binds directly to the Tata box while blank binds directly to the -10 and -35 sequences. For the first part, that is the TFIID, and the second part is the sigma factor. B says blank is a eukaryotic enzyme that links the 5' carbon of one nucleotide to the 5' carbon of another nucleotide, and that is the guanylyl transferase. C, blank uses the nucleoside guanosine as a cofactor during catalysis. I can't speak today. While blank uses adenine as a cofactor during catalysis. The first one is, and the second one is group 2 introns. D, blank is an endonuclease that assists in terminating transcription in eukaryotes. That is the CPSF. E says blank is a GTF, which acts as a bridge subunit to link TFIID to TFIIF/RNA polymerase 2. And then blank is a GTF, which acts to phosphorylate and activate RNA polymerase 2. The answers to those are 1, TFIIB, and for the second one, TFIIH. Then finally for F, it is asking, or it says the spliceosome is composed of subunits called blank. That one is the SNRNPs, or SNRs, and the second part says these spliceosome subunits are composed of 2 components. So that's talking about the SNRs themselves. It says one is proteins and the other is snRNA. All right, number 3. The question is, it says this question concerns the proteins involved in transcription and RNA modification. A, indicate 2 proteins in either prokaryotes or eukaryotes that act as helicase to separate 2 DNA strands during transcription. And so one of those is the TFIIH, and the other one that we found was RNA polymerase when it is bound to the promoter. B asks, indicate 1 protein in prokaryotes only that acts as a helicase to separate RNA from DNA during transcription. And that one protein is the rho protein. C, indicate 3 molecules in eukaryotes that function during RNA modification as ribozymes. The 3 that we were able to find are the SNRNP, the group 1 introns, and the group 2 introns. For D, it says indicate 1 protein in prokaryotes that acts as a transcription factor, and for that we put the sigma factor. And then finally for E, indicate 1 enzyme in eukaryotes that synthesizes a long string of identical RNA nucleotides, but does not use a template DNA strand for RNA synthesis. And that is our poly A polymerase. All right, number 4.
So for number 4, I'll just take this with me. Okay. So for number 4, the question is, the pre-mRNA for YFG is hybridized to the genomic DNA copy of YFG. So we have this pre-mRNA strand and we have these introns. The introns, after splicing, they get cut off and you get this hybridization going on. So I show that here in this image. For question B, the mature mRNA for YFG is hybridized to the genomic DNA copy of YFG. That's showing in this drawing over here where we have the mature mRNA going from 3' to 5', and then you have the DNA copy going 5' to 3'. Basically what's going on is you're going to have these intron loops being created where the introns were being spliced, and you have these R loops also occurring that will be going 3' to 5'. You have R loops being created where hybridization is occurring, and then you have binding occurring where the I loops are. And so in this, you're going to have 5 R groups and 4 I groups. For C, the question is, the mature RNA for YFG is hybridized to the genomic DNA copy of YFG. In this case, 5' most intron in the pre-mRNA contains a defective donor. So in this drawing, we have the defective donor right there. And so what's going to happen is that binding will be occurring. The I groups are only going to be occurring where the introns are being spliced, and then R loops will be occurring in between those. So we will have binding, intron loop, R group, intron loop, R, I, R. And for D, basically, the second and fourth intron in the pre-mRNA both contain a defective donor. And so 1, 2, 3, 4, defective, where they are defective, there will be binding, and where they're not defective and they are being spliced, you will have intron loops. And so in this, we will have 1 R loop and one R loop and 2 intron loops. That's
Okay. Number 5.
Sweet, come here. All right, so for number 5, A, which of the above is found in the bacteria E. coli? We got the rut, -35, and Noosa. B, which of the above is found in the yeast S. cerevisiae, or however you say that? We got -25, enhancer, group 1 intron, GC box, TFID, SNRNP, and silencer. And then C, which of the above is involved in transcription initiation? We got -25, enhancer, -35, GC box, TFID, silencer. And then D, which of the above is involved in the transcription termination? Got rut and Noosa. And then E, which of the above is a DNA sequence? We got -25, enhancer, -35, GC box, and silencer. So that's it. Let's see what happens.